everyone. Today on the WhiteOwl Talk Series, we will be interviewing someone who works in the very interesting field of paleogenomics. She's from New Zealand and now works in Sweden. She studied at the University of Otago, which is one of the top 1% universities in the world. She studied biochemistry, genetics, anthropology, and anatomy, and is now pursuing her PhD in the field of evolutionary biology. She extracts and maps mitochondrial DNA from animals that lived thousands of years back. If extracting base pairs from these DNA in these samples that have been exposed to elements of nature over thousands of years is not hard enough, she also goes on to analyze patterns to predict living conditions and reasons for going extinct for these ancient animals. So all of this sounds very complex, and I'm sure she can explain it much better than I can. So let's talk to our guest today, Miss Edana Lord, who is joining us from Stockholm, Sweden. It's great to have you here on the talk series. This talk series provides life lessons, which can help high school students make better life choices. So first of all, I want to congratulate you on publishing your paper, which talks about the cause of extinction of the woolly rhino.
Thank you.
So can you help us understand what paleogenomics is all about in simple terms?
So paleogenomics, or ancient DNA, so that's the study of DNA that has recovered from organisms long after they've died. So, for example, we can use chemistry methods to figure out how old samples are, and then we use more detailed chemistry as well to extract the DNA from that. And we're able to analyze it using lots of really cool methods, and be able to tell a lot about the population history and genetics and all sorts of things about those organisms.
Okay. How does a day in the life of an evolutionary biology researcher look like?
I mean, I guess we kind of have two sort of types of days. We can have lab days or computer days, I guess. So when we're in the lab, we're dressed up in these basically like spacesuits almost, but what everyone is sort of wearing at the moment, the PPE for coronavirus, that's what we use in the normal day in the lab. And it's not to protect ourselves, it's actually to protect the samples. So one tricky thing with ancient DNA is that the DNA degrades over time. So what we have to do is stop contamination from us getting into our samples, otherwise we have no data to work with. So we dress up in these interesting looking suits, and we're in the lab. We start basically sort of the process is we start with having a bone that's been, or a tooth or sediment even, that's been collected from an archaeological or paleontological site. And we take that out. We either, if it's a bone or a tooth, we drill it. So we collect a very small amount of bone, and then we make that into a little bit of powder. And then we use a bunch of chemicals to basically burst open the cells that are left in that material and extract the DNA out of that. And then we add different they're called primers or sequencing primers. So different sort of individual tags, DNA tags, to either side of whatever fragments of DNA are left in there. And that allows us to sequence them. We run these on these massive next generation sequencing machines. That's usually done outside of the lab itself. So a day in the lab is generally doing one of the many steps involved in that process. But then we also have all the data analysis that we have to do as well. So that's basically sitting at the computer, coding away, and running different types of analyses.
Which one do you prefer more, the computer part or the other one?
I think I prefer the lab. Yeah, it's a lot of fun in the lab, especially when you get to the stage where you're able to visualize whether there is any DNA in your sample And when there is, it's like, hey, it's really exciting. But no, I do enjoy the coding as well. It's certainly a very useful skill to have, to being able to do all these bioinformatic analyses. So I like the lab part, but I'm very keen to improve my skills in the coding section.
I was reading your recent study that was featured on CNN about a puppy from 14,000 years ago. So you had concluded a near perfect match with the Wooly Rhino DNA, which was found in the puppy. So how did you filter through the contamination and the degradation to arrive at this conclusion?
Yeah, so I'll give a little background on this sample because it's super fascinating. So this was a puppy that's discovered from a place called Tumat in Siberia. And what's really cool about this particular sample of canid is that it's a very well preserved specimen. So it has teeth and fur and stomach contents and all sorts of things. So it's been mummified essentially.
Yeah.
So then some researchers at the University of Copenhagen and also in Russia did an autopsy on it. And from that, they found in the stomach a small piece of tissue that has sort of skin and a little bit of sort of a yellow fur sort of on it. And because of the color of the fur and sort of, type of tissue, they thought that maybe it was a piece of cave lion. And at that time, we had a postdoctoral researcher in our group who was working on cave lions. So they cut a small chunk of tissue and sent it to our lab to confirm basically whether it was cave lion or something else. And so by doing that, you sequence the DNA from it, and then we use a pipeline where we map this DNA to the mitochondrial genomes of several different animals. So we have some that we use sort of for standard contamination checks, and that includes things like pig and cow and chicken, because these sorts of things can sometimes come in on the reagents that we use in our lab. But then we also, if we're trying to figure out the species, do a species ID of something, we throw in a bunch of other species. So for this one, we use like cave lion, woolly rhinoceros, woolly mammoth, probably like hyenas or all sorts of different mammals. So instead of it aligning nicely to the cave lion, the DNA sequences here are actually aligned to the woolly rhinoceros. So it's a woolly rhinoceros, piece of woolly rhinoceros in the stomach of a puppy. And the very, very cool thing about this is the puppy itself was dated to 14,000 years ago. And this lines up with the extinction date of woolly rhinoceros.
Oh, wow.
Yeah. So, of course, the first thing we had to do was check how old the actual tissue of the woolly rhinoceros in the stomach of the puppy was, because there is a potential chance. So around 14,000 years ago, the climate suddenly got quite warm. So there was a potential chance that this piece of woolly rhinoceros tissue may have actually been much older than the puppy itself, if the climate warmed and exposed a carcass that had been lying in the permafrost for thousands of years. So we wanted to double check that first. So we we dated the tissue itself, and that came back at around 14,000 years as well. So not only did the puppy eat a piece of woolly rhinoceros, but it also ate one of the last woolly rhinoceros.
But what I was confused about was that I was confused at how the puppy managed to eat a woolly rhinoceros.
Well, I mean, we don't know either, but we can speculate a little bit. So one of the options is that this puppy may have been part of a scavenging pack of wolves that may have either taken down a woolly rhinoceros themselves or found the carcass lying around and just nibbled on it a bit. The other interesting thing about the puppy is I don't think the researchers in Copenhagen have sequenced it enough yet to figure out whether it's actually on the wolf lineage or the dog lineage. Yeah. So if if it's one of the very early sort of domestic dogs, then there is also the potential of humans having a role in here somewhere. Did the humans take down the woolly rhinoceros and then feed it to the puppy? Or is it just something that's occurred in a more natural sort of setting? Unfortunately, we can't tell at this point. As far as I'm aware, there's no sort of injuries on the puppy that may suggest it got trampled by a woolly rhinoceros. I mean, we also don't know if the woolly rhinoceros tissue belonged to an animal that was dead or alive at the time of it being consumed. So there's so many questions.
In your PhD research, you joined two very fascinating areas, which are paleogenomics and climate change.
Mm-hmm.
How did you decide to use your background study at Otago to study ancient reasons for climate change?
Mm-hmm. So while I was studying at Otago, so I started with genetics, biochemistry, and all that you mentioned. And then for my master's project, I ended up delving into ancient DNA, looking at how humans transferred guinea pigs into the Caribbean and South America. And that really, really got me interested in ancient DNA and what I could do with that. So when I was sort of applying for PhD projects, I was keeping my options open. I knew I wanted it to be an ancient DNA, but it wasn't necessarily, didn't specifically have to be any one of the various options that you can do with ancient DNA. And then I came across this advertisement for a project here in Stockholm, looking at the impacts of past climate change on various fauna. And I was like, wow, that sounds awesome. That is something I could do and something I could see myself doing. So I guess the technical skills that I built at Otago allowed me to have the experience required in order to sort of jump track a little bit from the anthropological side of ancient DNA to come across into the more climate change aspects.
Your current position is the result of many years of studying topics related to biology. So when did you realize that you had a passion for biology, and did your high school support your interest?
Yeah, so I think I've always sort of had a passion sort of for biology and the natural world. I mean, I grew up watching a lot of the David Attenborough videos and things like that. And also Jane Goodall as well, seeing her. I remember very vividly going to a talk. She did a talk at the University of Otago and just being like, oh my God, you're amazing. This is so cool. But I did have sort of an interest in evolution from at least mid to late high school. We were doing stuff on human evolution, looking at the different fossils and things, and I think there was a few good discoveries around that time as well, looking at the Australopithecines. There we go. And also, yeah, things like that around human evolution was probably where I sort of started getting into more of the ancient DNA sides of things. But yeah, I know that my high school now is also very good at taking the students to public lectures at the university. So that's something that's really cool. They weren't doing that while I was there, but I have seen them since. So good on them for doing that. So any public lectures that are available, I would highly recommend people to go and watch them, because often they're sort of toned down a lot from big scary science to things that are a bit more digestible for a broader audience. So I started, sort of getting the interest there in high school. And then I remember vividly, my dad showed me a picture of all these biochemical pathways and how they fit together and all that. And I was just like, wow, that's amazing. I want to do that. That's really cool. And so I went to the university thinking to do a degree in biochemistry. And I had one semester of biochemistry and decided that I was no longer going to do a degree in biochemistry because that didn't actually sound like it was going to be as much fun as I thought it was. So I switched to genetics. And I also, in that year, took an archaeology paper. which is, I mean, a lot of people in ancient DNA come in with like, oh yes, I was, wanted to be Indiana Jones or whatever. But for me, not so much. So I came into the archaeology thing a little bit later. But no, continue to do genetics and bit of archaeology and anthropology and all that on the side, bit of zoology as well, just to keep all the evolution things in there, because that was really exciting to me. And then eventually, so I had gone to a few public lectures by who became my master's supervisor, Professor Lisa Metasue-Smith. And I remember when she was giving a lecture, actually in one of the courses that I took, she said, "Hi, I'm Lisa Metasue-Smith. I'm a molecular anthropologist." And I was like, there's actually a title for what I want to do. Molecular anthropologist, combining archaeology or combining the genetics with the archaeology and the anthropology. And so after that, I got in contact with her to do, firstly, it was an honours degree, which I'm not sure has an equivalent outside of sort of the Australia, New Zealand system, but it's sort of like a final year of a bachelor's, I guess. So I did that with her supervision, and then I went on to do masters with her as well.
And as you said, your job is difficult, but it's also a lot of fun.
Mm-hmm.
So researching the stomach contents of a 14,000-year-old puppy and concluding that it ate a woolly rhino as its last meal must have been really interesting. So what do you like the most about your job?
Oh, there's so much of it that I enjoy. I mean, getting to look at these animals that lived in the past, I mean, especially things that are extinct today, like the woolly rhinoceros, it's so cool being able to work on them and understand more about how they lived and what went on with them in the past. I mean, that is just super, super cool to do. Sort of kind of recreating, not really recreating history, but I think you know what I mean. Yeah. So being able to tell the stories of all these extinct animals and things that lived in the past is pretty cool. Obviously, yeah, I talked a little bit before about how sometimes we get things that are contaminated and they don't contain as much DNA as we think. So a lot of it is working with stuff that works.
Yeah.
So you can look at like 10, 20 bones or something and only find good DNA in one of them. Yeah. But one of them is enough in some cases to tell a good story, especially if it has very good DNA from the species that we're actually looking at, and we can maybe sequence the mitochondria or the nuclear genome as well. So I think the exciting things is working with these extinct animals and also getting really excited when it works. Yeah.
The current tools for DNA replication, like you said, was NGS. Are compared to the PCRs, which were used in 1990s, much more advanced. Mm-hmm. So what kind of breakthroughs and discoveries do you expect to make over the next 10 years with these tools?
So I think one of the big things is that sequencing is still getting cheaper and easier to do. So I think even just recently they've sort of decontinued a whole sort of set of sequencing machines that were very, very prevalent up until a couple of years ago and superseded them with things that sequence billions more DNA reads. So DNA sequencing is still improving. We also have other types of sequencing. So there's been a couple or a few papers in ancient DNA done with, I think, single cell or single molecule sequencing, which is also really, really cool. Other sort of massive, maybe not necessarily massive, but other sort of cool things that are pushing the field is all the bioinformatics analyses. So developing all the statistical frameworks behind these analyses to be able to work with not maybe like 10 or 20 genomes, but to work with hundreds and thousands of genomes. So this is sort of stuff that's being done in sort of, I guess, the modern genetics fields. And it's definitely, as we are able to sequence more and more samples in ancient DNA, is also something that will then filter down towards ancient DNA. The other big thing from a methodological standpoint is improved methods for extracting DNA, especially from smaller and smaller amounts of material. So when the field sort of first became a thing, you kind of had to use a good chunk or so of bone, but now we're down to sort of about anywhere between sort of 2 and 50 milligrams or something of bone. So it's becoming a lot, you need a lot less now, but I think there's still room for improvement there to really be able to get all the DNA from that sample out.
Yeah,
and sequence
a lot less laborious than 1990s, I think.
Mm-hmm.
Yeah, and do you think these tools can help in other areas such as forensic?
Absolutely, yeah. So ancient DNA and forensics has a lot of crossover with methodology. Definitely.
For high schoolers trying to find their passion, what advice would you give them?
Follow the things that interest you. I think we've all had those sort of moments where you just can't explain why you're so excited about something. And I think that sort of a passion is something that you should follow for sure. Though I think I would also say that sometimes it's nice to have like multiple passions, ones that you keep as hobbies and ones that you want to invest in for a career. But no, follow your heart and your dreams, I guess, would be that one. But
how would you say that someone would find their passion? Because it's difficult for people to find what it is they like to do.
Yeah.
So how do you think?
I mean, if you're sort of in the science or whatever, there's a lot of like YouTube videos and things out there now with lots of different scientists and things talking about their work. So there's that. Getting onto sort of public lectures as well is very cool. Yeah, talking to your teachers at school and things, they can also point you in the direction of maybe where to find more information if you're sort of excited about something or want to learn more. Of course, there can be a lot of misinformation on the internet these days, so you have to be a little bit careful sometimes. But yeah, talking to people, teachers, parents, getting their sort of, or like bouncing ideas off them, being like, I'm kind of interested in this. What could I do with it?
Yeah. So that's it. Thank you for speaking with us today. And congratulations again on your paper. And we look forward to reading about many more fascinating researches coming out of your lab. And we wish you the best with your doctorate.
Thank you. And thank you for reaching out. It was really lovely.